Students, we are going to study Nucleolus today. This Nucleolus was discovered first of all by Fontana in 1870, and it is found inside the nucleus, and it appears as if it's a membrane bound organelle, but it is not so. What it is, let us see. It is stained by pyrrole envy, spheroidal within the nucleus. It may be central or peripheral in its location. It is found in close association with the nucleolar organizer region of the chromosomes, and it is present in the actively synthesizing cells. The cells which are very active in synthesis, they have very large nucleoli. Let us come to the structure of the nucleolus. It has matrix which consists of large number of scattered granules and fibrils. Inside the matrix we have chromatin, which is DNA, as you know. It is fusion positive and it serves as a template for formation of 45S RNA. Surrounding the nucleolus like a shell is a perinucleolar chromatin, which may be in form of wall, and it gives the appearance of a membrane to the nucleolus, which is in fact is not there. Then we have fibrils. These are 80 to 100 nanometers, which are precursors of granules and contains RNA. That means the chromatin gets itself converted into RNA, and these fibrils, which are cited in the matrix, are nothing but they are RNA. Then we also cite some granules which are ribonucleoprotein particles, and they have dimension of around 150 to 200 angstroms. These granule contains proteins and RNA, which are 25 Sidberg RNAs. These granules are actually the precursors of ribosomes. Let us see the functions of nucleolus. It is RNA production. As I have mentioned right now, produces 70 to 90% of the cellular RNA, and it is the source of RNA. Chromatin in nucleolus is nothing but DNA, which codes for RNA, and granules are actually the precursors of ribosomes. So how does the formation flows? Chromatin, which is rDNA, and then gets after transcription gets converted into rRNA, and these rRNA they now get converted into ribonucleoprotein particles, which are the precursors of ribosomes, and these RNP particles they ultimately get converted into ribosomes. Second function is protein synthesis. Ribosomal proteins are synthesized in nucleolus. Then the prime function is formation of ribosomes. As I have told you, DNA serves as template for 45S and 25S Sidberg rRNA. Half of it gets degraded, then it gets converted into 18 Sidberg and 20 Sidberg S rRNA, then which forms two subunits of ribosomes, 40S and 60S. See the structure of the nucleolus. The outer part, which is shown in red color, is DNA, and the RNA precursors inside this in the matrix are RNA fibrils, which are rRNA fibrils. Then you can cite the granules of black color, which are precursors of ribosomes, and inside you can also see intranucleolar chromatin. perinuclear chromatin, which is rDNA, and fibrils are precursors of granules, and granules are ribonucleoprotein particles and are precursors of ribosomes. So this is a small chapter. Thank you so much, students. In the next lecture, we'll study endoplasmic reticulum. Thank you very much.